Listen, the talk is going to focus on two topics, privileged and contingent, right? So privileged are things that cannot not happen in an environment, and the contingent are things that might happen or might not happen because of a historical accident. To show you how I have basically wasted an entire career on this, this is an article that I wrote with Andy Ellington in 1988 saying, titled The Purpose, Interpreting the Behaviors of Life, Purpose or Pedigree. And you can see how this has been a long-standing discussion. So there are natural ribonucleoside triphosphates. The triphosphate part is in magenta. The question you're supposed to ask are these structures privileged? That is, would they have the only ones that could possibly do what with RNA, what RNA is supposed to do, or are there others? Or are these historical accidents by which other structures are possible? Of course, as a synthetic organic chemist, I am able to ask the what if and why not questions, and so these are other ribonucleoside triphosphates that are possible. For those of you who remember your basic biochemistry, these are base pairs that are held together by Watson, Crick, Franklin, Pauling, Chargaff, Donahue pairs. I don't know that I've got them all, but they're certainly two complementarity rules. One is that big pairs with small size complementarity, so large purines pair with small pyrimidines. There's also a hydrogen bond complementarity in these things, which hydrogen bond donors pair with hydrogen bond acceptors. So you can make 8-letter RNA, 10-letter RNA, 12-letter RNA. They are 8-letter RNA may even be better, right? It's storing 3 bits of information per site, whereas 4-letter RNA only really stores 2 bits of information. And so you can make an argument. So these are genetic systems. They can contain information. They still do Watson-Crick base pairing in the way that you're familiar with. Millie Georgiadis, who's a colleague of ours, has done crystal structures of these, shows they're still forming double helices. So they're a genetic information system. They're also evolvable. You got to keep in mind that for Darwinian evolution, it's not just that you can have a molecular recognition. You have to be able to change the building blocks to change molecular recognition without destroying the rules by which molecular recognition occurs in the same time. So there is something called the polyelectrolyte theory of the gene, which says fundamentally that in order to support the Darwinian needs, informational needs of Darwinian evolution, the biopolymer, the informational biopolymer, which must have a repeating charge in the backbone. And that's because that repeating charge will so dominate the physical properties of the molecule that you can change its information content by exchanging the building blocks without having the thing precipitate. So proteins, for example, do not have this particular property. So the informational units also must be size, shape, interchangeable. That's a component about homochirality as well. And that's a comment that goes all the way back to Erwin Schrödinger and his aperiodic crystal structure. So that's great. So this is also an evolvable system, and I'm not going to have a chance to talk much about this in the sense that we have, of course, getting it evolving in the laboratory. This is a case of a sequence that has, as you can see, 6 different DNA letters. It's something that binds to liver cancer cells and helps us deliver drugs to them. And so that all the students in the room should know that the most important thing in science is money. This is what pays the bills. It's not prebiotic chemistry, as it turns out. So. And it's anthropogenic because, of course, it was created by people, anthropos, and there's a number of people who are very important in this. So Tillman Krauk, for example, you'll see a base pair, which is K. That's named after Krauk. Andy Ellington, I just mentioned, he wrote the article together. He's now a professor at the University of Texas, and various other people have contributed to them. And I'll show you their pictures as we go through the talk. So please, I'm going to ask you to remember what is privileged and what is not privileged versus contingent when we go off to Mars to seek life living there today. Because we now know that GACU are not the only base pairs able to support evolution. Martians must be able to do Darwinian evolution, that sort of theory. We think that Darwinian evolution with non-prospective mutations is the only way that matter can organize itself to give properties that we value in life. We think, therefore, that Martians must have an informational biopolymer to support this. It may have RNA, but it may have different molecules or different building blocks in RNA, but it could easily have bases other than the ones that we have: adenine, uracil, guanine, and cytosine. In fact, some life on Earth does not use G, A, C, and U. Some viruses that infect cyanobacteria have a different A version. So we're going to have to worry about this when we go looking for Mars, because we cannot just necessarily do PCR of the Martian surface using the assumption that the Martian nucleic acids are just like Earth nucleic acids. Of course, we also have to worry about what metabolism would make Martians fit for Mars. Remember, Mars has evolved as well as Earth in terms of its planetary environment, and so life would have had to have found a way to survive that planetary evolution. But whatever informational biopolymer Martians have, it must have a repeating charge. So hold that thought for a few slides. So now let's talk about geology, rocks. All right, so you take ribonucleoside triphosphates and you mix them with rocks. Now, Bettina gave you an excellent introduction. I'm not going to try to reproduce that. But she told you about basalt, and she taught you about basalt glass. So one of the questions is, what happens when you put these triphosphates on basalt glass? And this was a paper that was published about 2 years ago, and these are the people: Elisa, Hyojun, Craig, and Sue Wei were the people who did this work. And what you get out of that is RNA. Now, I call it polyribonucleic acid because we don't actually know 100% of what the backbone linkages are, but we do know something about that, which I'll tell you in a minute. So you get RNA, there it is. There's the triphosphates that you put in radioactively. There is the RNA that's coming out of it. You can follow the kinetics of formation. Over about a month, about 0.5 milligram of RNA is made for about 1 kilogram of basalt. That is, in the technical term, a shitload. Okay, that's a lot of RNA, okay? And so it's a privileged molecule in the sense that if you have triphosphates and you have basalt glass, you cannot not have RNA. Now, different glasses are different. So quartz glass does not work. Felsic glasses, which are presumably later in the history of evolution of the planet, don't work as well, but they do work. And of course, the glass that we got to use this in particular was collected from Iceland. And Bettina always asked me whether I got a permit to collect the glass, and the answer is yes. We paid the Iceland government money to get that material. So there you go. So now the question for all of you who are not in this field is very simple. It is sort of na und, so what? All right. Fair enough. So the answer is, why is formation of RNA on basalt important? And the answer to that is briefly because RNA can do both genetics and catalysis. Now, you're quite familiar with what RNA does by genetics. If you've had COVID, it's using RNA as its genetic material. It's capable of supporting evolution. But of course, ribosomal RNA, which is the orange parts of this ribosomal structure, is actually the catalyst that makes proteins in all living organisms that we know on Earth, including yourself. And so this implies, and this working backwards in time, it gives the impression that we all descended from an RNA world which preceded protein translation, which is an episode of life on Earth where RNA did both genetics and catalysis in a one biopolymer life form. That is, the only genetically encoded component of biological catalysis was RNA. And I don't know how many of you read the side of your vitamin pills, but you can actually see vestiges of all this. We're talking about molecular fossils. So, if you read the back of your pills, you have thiamine and riboflavin and niacin and, of course, various other compounds here. I've just drawn nicotinamide adenine dinucleotide. It is actually the active form of niacin. You have a piece of RNA stuck onto the niacin, and that's what does oxidation reduction reactions in your body. The same thing for flavin adenine dinucleotide. The riboflavin, which you are eating in your vitamin pills, 17 milligrams, 100% of your dietary requirement, is stuck onto an RNA molecule. And this goes back a long time, but the idea here is that these are vestiges of an RNA world where those RNA pieces, our handles, are put onto molecules that have the chemical reactivity. The RNA part doesn't actually contribute much to the chemical reactivity, but it does allow the system to bind in particular to proteins today, but the argument was that it emerged when the RNA was the only molecule that you had available. So these RNA cofactors arose in the RNA world to support complex RNA-catalyzed metabolism in 1989. Good God, almost a quarter century ago. Andy and I put together a complete metabolism based on an analysis of what was then known about modern protein sequences to infer the metabolism of the last RNA world organism and the first organism actually to invent translation. So that's kind of cool, and that's the argument for this, and that's sort of the argument that we make. Actually, you saw some of this from Georg this morning, right? We're looking at all modern species. We're making inferences about the presence of ancient forms of ancient molecules. So the modern species all use nicotinamide adenine dinucleotide. Therefore, the ancient form did as well. Some of these ancient elongation factors that are involved in the ribosomal translation actually have been resurrected. But the argument is that this is the case where RNA world existed. These are the evidence from the modern biochemistry that there was an episode of life on Earth where RNA was the only genetically encoded component of biological catalysis. Now, it's much more controversial to say that that RNA world came from a prebiotic soup where RNA was made first, and of course that's why it's important for RNA to be formed on basalt rock or something else that's plausibly geological. So why is it surprising that you can get RNA? And the answer to this comes from a long history. It includes Jerry Joyce and Leslie Orgel. They're famous for having referred to RNA as the prebiotic chemist's nightmare. And if you actually look at this structure, all the bonds in color, the color doesn't come out so well here, are bonds that are thermodynamically unstable in water. I put some rate constants up there, so the half-lives are somewhere in the neighborhood of 10 to the 10 seconds. So, you know, it's maybe 300 years, so it's not all that unstable as these things go. But still, if they're thermodynamically unstable, you have to work to get them to form in water. And that's actually a central paradox, because RNA works in water. It's a polyelectrolyte. As we have said a moment ago, according to the polyelectrolyte theory of the gene, you have to be able to have a repeating charge in order to meet the informational needs of Darwinian evolution. And that really is a molecule that does not work in hexane. It works in water. So, but RNA is unstable in water, and so the implication it can't be made in water, and so you have the formation of a logical paradox. It must work in water, but it can't be made in water, therefore it can't originate. So that's kind of a problem. And so there has been this elephant in the middle of the room in the whole field of prebiotic chemistry, right, where all sorts of people work on RNA pieces. Thomas Carell, I rely on you guys to tell me what to think. There's also a whole group of people over here. Jerry is one, Jack Shostak, Eric Hyde, and Irene Chen. The Simons Foundation funds it, which are showing that RNA can evolve, but this step, going from the pieces to the RNA, really has been a system which we don't dare to work with. And so this becomes very important, that you can get basalt to get that step to go from the pieces to the rest of the molecule. Now, the problem is that nobody believed us, okay? Actually, extraordinary claims demand extraordinary evidence. Keep in mind that if I'm saying that I can just take basalt rock, which everybody agrees is abundant on Earth, basalt glass, and just spoon a little bit of triphosphate on it and get 150 mer RNA molecules, which is longer than what we think is necessary to get replication out of this system, I damn well better get the structure right. I'm not sure how that's going to get translated into German. Never mind. So let me give you a bit of pieces of structure proof, okay? So right there is the RNA, there is the triphosphate. I'm going to join RNA. There's the two OH groups on, and this is the bond that's going to be formed. Okay, we can put radioactive material, that's that radioactive phosphorus in red, onto C, which is going to be joined to a base U right in front of it. It could be any base, but this is just the example. So when you do that, you make a bond that joins that radioactive phosphorus to the blue U and to the magenta C. That's a connection. It's a covalent bond. Jerry, to this day, still will not concede to us that we can make RNA on basalt glass, but this is the evidence, okay? So now we can break this RNA molecule apart in base degradation, and that breaks this bond, which leaves the radioactive phosphorus on the blue U, the radioactive phosphorus that started on the green C. So that movement of that phosphorus from this molecule to that molecule is the evidence that these molecules were not in some non-covalent aggregate, but rather the bond was there. We can also go back and degrade that bond by enzymes like ribonuclease T2, which, ribonuclease 1 is what's being used here, where we can actually break down, and Elisa Biondi went to great work to show that these 2' 5' linkages are not degraded. The 3' 5' linkages are degraded. So we know that maybe about 80% of the bonds are the, quote, natural 3' 5' linkages. And when I say natural, that means what's found in your body. So the question you could ask, well, why the hell did Benner go into the lab and put triphosphates on basalt? Well, Bettina has already given you the answer to that question, right? Planetary conditions drive prebiotic chemistry, especially the part that is privileged, the chemistry that cannot not happen. And so there are many questions about early Earth, Mars, how much dry land there was. You just saw a slide of that. We don't know what the precise solar fluxes were. There's all sorts of things. But we actually do have samples of the Earth of that antiquity. And Didier disparaged these poor little zircon crystals. These are wonderful crystals, right? This is work from Dustin Trail at Rochester. Right, you can get these crystals, you can go in, they absorb uranyl cations, uranium-235, 238. There are 2 independent decay series to lead where you can date these crystals. Those 2 dates have to be coherent so that you assume that the crystals have not been disturbed in the last 4 billion years. He's measuring the rates of cerium-4 to cerium-3 in those crystals. He is doing experimental mineralogy. He actually grows crystals of zircon in his laboratory from mantle analogs, which have different redox states. And he can say that, yes, this is roughly the phthalate magnetite redox state, which means that hydrogen is coming out as water, not as H2. Carbon is coming out of this mantle as CO2, not CH4 and other things. And of course, there are your energy sources, and there's the lightning that we actually love to have around volcanoes. It's a source of energy. So why the hell does Benner keep talking about Mars? Well, the answer to this is because Earth may not have had so much dry land, as Dimitar has said, as we have said. We're looking for these intermittently hydrated, constrained aqueous aquifers where chemistry can be done. They have to have access to the atmospheric input. We don't want it diluted into a global ocean. There is what primitive Earth looked like, except for that guy here. That's an Icelandic basalt where we picked up these Icelandic basalt lava flows. There's a basalt on Mars. We know that Mars had dry land for sure. We're not all that certain about Earth, but all the chemistry I will tell you about will be done on Mars as well as on Earth. Another tidbit, right? Borate, H4BO4-, does not inhibit RNA synthesis on basalt glass. So why is that important? Well, the answer is because borate binds to organic molecules with adjacent OH groups. If you look at this condensation, you see 2 adjacent OH groups. We have to worry about whether borate will bind to those 2 OH groups and form a inhibiting complex and not allow the formation of RNA to occur. It turns out that borate actually helps basalt catalyze the formation of RNA. We think that's a pH effect, actually, but never mind. And borate actually helps make triphosphates. If I have a nucleoside like this guy, and I want to put 3 phosphates on it to get a triphosphate, and if I put it in with nickel, which is coming, you'll see in a minute, from meteoritic cores, you make phosphates, but you put phosphates on every one of the OH groups. In contrast, if borate is present, borate loves to condense to 2 OH groups on the bottom. It does so, forms a complex that blocks the OH groups, and you get, as privileged products, 5' phosphorylation. So borate's actually quite useful. So RNA cannot not be formed on a surface of a rocky planet if it contains borate and can make triphosphates. Of course, did the Earth not have borate? Well, there's a big fight over this. I'm not going to go into great detail. Bob and Ed were worried that there was not enough plate tectonics to fractionate the environment to get scarce borate concentrated. We actually argued that it was being concentrated in residuous igneous melt and then being concentrated further in the hydrosphere. This case was settled when they discovered a lot of borate in the surface of Mars, which did not have all that much plate tectonics. So Bob Hazen now says, I can have any mineral I want, which is really quite sweet of him. So borate also manages carbohydrate reactivity. So Stanley Miller wrote a paper in 1995 saying that because of the instability of these carbohydrates like ribose, they cannot be prebiotic reagents. And you just heard of the discussion section this morning. People say, well, carbohydrates all stable, they have to go away. Well, that's the so-called tar paradox, as I like to say, right? You're looking at sugars where if you heat them up, they form caramel, and they do that by these enolization processes. And that's the so-called tar paradox, because if I write down every enolization and aldol reaction that can happen in this system, and I mean, I just stopped here arbitrarily. This could go on. This is what caramelization looks like. It's all of those reactions. But if you look at those molecules carefully, you'll notice that they have adjacent OH groups everywhere. And in the presence of borate, that controls this system to that box. So you make a branched 5-carbon carbohydrate. We're talking about how glucose and fructose were what people think of as carbohydrates. You can't make them by this cycle in the presence of borate. All the carbohydrate goes to that species, which forms a borate complex, which is stable for decades. It is hard as a rock. In fact, the only thing wrong with it is it's very hard to get it to rearrange to its isomeric form, ribose, the R in RNA, without getting rid of the borate. So what carbohydrates were not present? Well, the answer is we actually have atmospheric models working with CO2 where you make formaldehyde and maybe parts per million glycolaldehyde, a lot more formaldehyde. You also have the volcanic material I've already shown you with sulfur dioxide emerging from the volcano. It turns out that if you actually measure the amounts of material you're making, about 10 to the 21 molecules per square meter per year. That's about the same amount as the sulfur dioxide that is emerging from volcanism, I guess. It's a good question. Those two react to form a bisulfide addition product, and those materials are stable because they do not have C double bond O groups. And these could not have been raining out on the surface so that you would have a metric ton of these materials in this room after a year based on known photochemistry. So saying that Earth did not have carbohydrate reservoirs is like saying that Titan, which has a much more reducing environment, does not have acetylene polymers on its surface. It is. The whole surface of Titan is gooey with organics, cold organics. Earth was gooey with these stabilized materials, and what they did under borate constraint is filter these pentoses. 5-carbon species is the endpoint, not 6 carbons, not 8 carbons. Okay, so that's great. So privileged chemistry here, privileged chemistry here. What about this? Well, I'm not going to go to great length. We've already heard now from several speakers that the FMQ, the phthalate magnetite quartz, redox potential from the mantle is not producing a lot of reduced nitrogen. But that is a model that's too placid. There certainly is bombardment of, say, Vesta-sized or Ceres-sized asteroids, which have their own iron cores. They certainly would reset mantle clocks at the 10 to the 25 gram impactor scale. They certainly would have killed off whatever life you already had. They had their own iron nickel cores, and they certainly would have reduced the atmosphere. And I'm not going to tell you about what happens here. I rely on Tom Carell to tell me what I should think about this. But the point is, this chemistry can be privileged as well. I mentioned this to Kevin Zonley in 2019, and he came up with these nice models. You got an impact of a Vesta at time 0. 500,000 years later, you got the time for doing productive formation of reduced base compounds. And as you heard before, so what you have on Earth is more and more dry land. That's important. You have, of course, the carbohydrate is building up on the surface. You have, of course, the reduced nitrogen compounds only forming right after the impact. And so you have a probability maximum for when RNA life could have emerged based on this combination of chemistry. And it's about 4.3 plus or minus 0.05 billion years ago. It was on a Tuesday. Okay, so that's when this happened. That's when life emerged. So under this model. So you're looking at all of this privileged chemistry around the perimeter, of course, we now had to fill in the middle. I have to thank Ram Krishnamurthy and Albert Eschenmoser for the chemistry to fill in the middle. It turns out that they have a molecule, diamidophosphate. It's not quite so privileged for the question I just asked you. I don't quite understand how I make the connection out of basalt rock and phosphate that gets there. Ram keeps coming up with good ideas as to how to improve this, not the least of which is the reaction of reduced phosphorus in meteoric iron core schreibersite to form this particular molecule. This molecule is not quite as high yielding as we like. I mean, we say 17%. That's a great yield for prebiotic chemistry, but it's not good enough to satisfy Dimitar, nor should it be. Yes, and so the bottom line is that's why this is not quite so privileged. But there is the mostly privileged model, right? A large amount of carbohydrates being stabilized as bisulfite addition comments. You may have John Sutherland has other chemistry, other people have other chemistry. I have opinions about that, but for this chemistry, it happened. It could not have happened. And so you have these reservoirs of carbohydrates on the surface, and you have basalt. We have to understand more about how phosphorus enters that system. But if it does, in the presence of borate, you have oligomeric RNA without any trouble. All right, so there's the problem solved. Well, the answer is not quite. There is this question of chirality, and it's right in the center of this plot. You have two ways of getting from this branch pentose to ribose. One involves molybdenum-6 rearrangements. If that worked, you have to solve the chirality problem here, not earlier. All the enolizations will destroy any chirality solution you have earlier. Not later. That center is fixed, and it will be propagated throughout the rest of this slide. It's got to be solved at those magenta compounds. If you don't have molybdenum, you have to do a retroaldol reaction. These molecules are flat. You've lost the chirality. You have to solve the problem there at the aldol condensation. We have not bothered to look at whether the borate complex of this crystallizes as a conglomerate or whether it crystallizes as a racemate. We should obviously do that tomorrow. So, will that solve the origins of life problem? Well, the answer is we see it as shifting the bottleneck. It's removing the element from the room. So previously, prebiotic formation of RNA was truly paradoxical. Now it can be the focus of hypothesis-driven research. The other problems now take place as bottlenecks, compartmentalization, which you may hear about later today, the poor catalytic power of RNA, using the model to search for life on Mars and Europa. Now, the poor catalytic power of RNA may be a reason why the RNA world invented proteins. But RNA has other ways to improve its catalytic power, and I've actually already shown you these. You can expand the alphabet, right? So remember, before I do that, we have to ask the question, what metabolism would make Martian life fit for Mars? That's a very good question. So if you give me a planetary environment, I will give you a few metabolisms fit for that environment, unless it's Laura who gives me a molten magma ocean, which I'm going to say you're on your own. So this is because magma is not a good solvent for life, as far as I know. But we've done Venus. We can certainly do Mars, and Mars is kind of nice because Mars has abundant solar energy and it has abundant CO2. What is scarce is hydrogen atoms with electrons. So if you were going to sit down knowing what you know, what kind of metabolism would survive on Mars? Well, it might as well use bacteria, okay? It's going to be an auxotroph. It's going to use the CO2 and solar flux to make biomass. It's going to be respiring. That is, in the evening, it will have overnight metabolism where it burns the biomass, just like the plants outside. But unlike the plants outside, Martian life does not have access to much atmospheric O2, so it will store the oxygen that it makes during the day for the respiration at night. And if you look closely, that has the convenient acronyms of a bacterial auxotroph respiring by, it's an autotroph actually, respiring using stored oxygen for overnight metabolism, which happens to have the acronym Barsoom. Now, oxygen storage is the principal difference, and so if you go to a Martian bar at night, you'll notice all the Martians will be carrying the oxygen with them that they made during the day because they are trying to survive in an atmosphere. Now, by the way, in case you didn't know, Barsoom gives all of the Viking results in 1976. You saw this. You saw fixation of carbon dioxide in the Viking 76, you saw the respiration, and you saw the release of stored dioxygen. Okay, so you can ask the question, why the hell does nobody in this room know that we discovered life on Mars with this metabolism in 1976? a very good question. And Thomas suggested I redo my entire talk to just focus on this one issue, which I probably should have because it's more interesting to the students. The short answer was that they misinterpreted a Viking gas chromatography mass spectrometry experiment to conclude that there are no organics on Mars. Now, of course, my only contribution to this was this paper which we wrote 25 years ago, pointing out that GC mass spec could not have detected the dominant organic molecule on Mars had it been sitting on a pile of them. Nevertheless, the culture was set. So Vicky Meadows and Heather Graham have a report in 2022 where they actually repeat the statement that there are no organics in the soil of Mars, even though rovers have found them all over the place. It's a remarkable statement about how a wrong experimental result has poisoned the culture so much that you don't actually have the dialectic established, what should have been established after the Viking result, a pro-life community, an anti-life community. Jerry Soffin stepped in and said, nope. Jerry was the head of the Mars Viking mission. No organics, that's the ball game, no life. And that result was wrongly interpreted there. Of course, there are organics. So, of course, Mars could have, of course, solved its phenotypic problem, that is bad RNA catalysis by expanding the genetic alphabet. I'm running out of time, but I'll just show you some more results from Elisa and from Tue and from Craig, where they have actually shown that if you can go to a 6-letter alphabet and evolve catalysts, the catalysts coming out of a 6-letter alphabet random RNA sequence are on the order of 100,000-fold better than the catalysts as you're getting out of a 4-letter alphabet. So it's possible that when we go to Mars and look for life, that's a problem. We don't want to necessarily look for RNA exactly like what we have in our own bodies on Earth, but we still have to remember that whatever informational biopolymer the Martian system has to support Darwinian evolution, which is a universal, must also universally have a repeating charge in the backbone. It must be a polyelectrolyte. Now, what's nice is that polyelectrolytes are easy to concentrate from dilute solution. And so there's electrodialysis. It's a machine that we are building today with NIAC, NASA support. And so the idea is very simple, right? I'm going to sit on Mars. Elon Musk will be pumping a large amount of water to make propellant for human missions to Mars. We're going to sit astride this with a device which looks like this. Oops, sorry. Let's try that. And what's going to happen is particles of Mars are going to flow through it. Every now a polyelectrolyte will come from a Martian life, and that will go in the direction of the anode if it's a polyanion. Of course, if Martian life has a different history, natural history than life on Earth, it could have cationic DNA, and of course that will be separated toward the cathode. But in any case, we can concentrate very sparse genetic material from very large samples of water using this particular device. Well, I'm out of time, but I have to make sure that you remember something from the talk because it's been a complicated talk, right? So what you've seen is a possible solution for the bottleneck paradox in the RNA first model for the origin of Earth, and that is privileged chemistry, most partly, that makes RNA on basalt glass. Borate privilege is a lot of other parts of the chemistry, including the phosphate formation, as well as the carbohydrate, controls the carbohydrate instability. The reducing atmosphere privilege is the formation of nucleobases. And from impact history, this process is going to have occurred optimally or most likely at about 4.3 times 10 to the, well, 4.3 billion years ago, plus or minus a couple 100 million or 50 million, something like that. This chemistry works both on Earth and on Mars. But where Mars we have more dry land. So we've shifted the bottleneck problem. Now the problems are compartmentalization or maybe poor RNA catalytic power. That might be able to be overcome by inventing proteins. This is what your ancestors did, but it also can be overcome. The poor catalytic power of RNA might be able to be overcome by alphabets that have expanded and therefore building blocks and therefore are richer as biocatalysts. And so then we can use what we have learned to seek life in the cosmos. Again, I'm looking for anybody who has a planet who wants to have a hypothetical metabolism on that planet for fitness. But it's got to keep in mind that Mars may have a different type of DNA, but it must be a polyelectrolyte. We have a way of actually finding that universally, and it's being built. Let me just close with one final slide and one comma one observation. This is the big picture of what we do in our laboratory, including working backwards in time to a minimal life to try to understand the essence, trying to create our own form of life that's playing God. There's the big picture, which I've told you about, which is trying to get life to originate from privileged chemistry. And of course, there's exploration. But Georg made an amusing remark this morning. He pointed out that there are all kinds of enzymes out there, all kinds of biological phenomena, and almost none of them are being studied from the perspective of natural history, right? The fact that I can write with Andy Ellington this article in 1998, and then 30 years later, in 2017, basically write the same article with the title Uniting Natural History with Molecular Sciences, the ultimate multidisciplinarity shows how resistant it has been to get the natural sciences and the physical sciences to come together. So you have all sorts of people starting new institutes at Heidelberg for life detection, and DDA has one, and Harvard has one, right? They don't have hardly anybody who is a physical organic chemist working with them. Well, Ram, I guess I'm going to call you. That's the highest compliment I can give somebody, as you know. But almost, for the young people in this room, almost nobody has made this jump. And the place is just, the natural world is rich with target-rich opportunity, as we like to say, for people who are willing to say natural history on one side. But remember, most physical scientists don't think evolutionary organismic biologists are scientists at all, right? They think this is just foo-foo, right? But just be able to join these two is an opportunity that has remained for 30 or 40 years now, and it's an opportunity for all the young people in the audience to consider as a way of actually breaking through into territory that has not yet been covered. So with that, let me thank you for your attention. I'd be happy to answer any questions.